Alright, AP Bio, here we go. I'm going to post this video to YouTube as well, and I'm going over the DNA replication graphic organizer as well as pulling from the PowerPoint Chapter 16, DNA Discoveries, Simple Replication 2018. You can find that on Schoology. I'm going to just go through a couple images because you can't see some of the things that I would be able to draw online. So one of the first things is going to be down here in this corner, the subunit of DNA. It's called a nucleotide. As a subunit, it still has three parts to it. So I'm using three colors here for the sugar, the phosphate, and the base. And remember down here, this is not DNA, deoxyribose sugar. This is actually an RNA base because it has the oxygen for ribose sugar. So go ahead and cross off that O to confirm that this is a DNA base for deoxyribose sugar. So we got a sugar in the middle with carbons numbered 1, 2, 3, 4, 5. I'll talk about that. The nitrogenous base, which you're probably familiar with, A, T, C, and G, and a phosphate. So all three pieces of the subunit are considered the nucleotide as one. You are probably familiar with DNA bonding, the complementary bonding, A to T, C to G. This is what makes things just right. Over here I've got purines and pyrimidines. Probably going really fast. Sorry, mine's all written down. Purines have two rings in their nitrogenous base. You can see an A has two carbon rings, a G has two carbon rings, and pyrimidines have a single carbon ring structure. That's the C and the T, cytosine and thymine. So with DNA bonding across the backbone, the hydrogen bonding across is going to interact with like the nitrogens, the hydrogens, the oxygens of these bases. And C to G, this pyrimidine to this purine, it's going to have three hydrogen bonds. This purine to this pyrimidine, A to T, is going to have two hydrogen bonds. And if you go horizontally across the backbone, you've got 1, 2, 3 rings. 1, 2, 3 rings. When DNA is proofreading itself, if a T and a C are trying to be bonded together, the backbone divots in. It's too narrow. And if an A and a G try to be paired together, we've got 4 rings across, we got a bulge and it goes too wide. So when proofreading enzymes are running down the DNA double helix, they're looking first to make sure that it's just right, the same distance across the whole time, meaning the correct complementary bonding was done. When you get a T and a C, it goes too small, A and a G, it goes too big, and these are going to get edited and fixed. So over here, you've got kind of everything in detail with all the atoms. So across the backbone, it's going to go phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And the other one, same thing, phosphate, sugar, but they're running in opposite directions. Let me show you something off the PowerPoint for that. This might be new to you. If you've taken genetics, you've probably heard of the 5' to 3' directionality of DNA. If not, pay attention, okay? So when carbons are getting numbered in the, touchscreen, great. All right. When carbons are getting numbered in the ring, we start here with carbon number 1, carbon number 2, carbon number 3, number 4, and the carbon number 5 is actually outside of the ring. This is not like glucose. The deoxyribose here is 5 sides, but then the 6th carbon is on the outside. So there are 5 carbons in this sugar structure, and when it's in the entire nucleotide, here we go. We've got our base that is attached to carbon number 1 every time. 2 doesn't have anything attached to it other than this hydroxyl. 3 is where bonding is going to occur southward, let's say. 4 can't do any more bonding because it's connected to 3 and 5, and 5 is connected to the phosphate where we're going to form our other bond. But when DNA nucleotides get connected to each other, every single time the enzymes in all these organisms' bodies that are replicating, building DNA, they can only attach in this direction, going from 5' to 3'. Next one, 5' to 3'. Next one, 5' to 3'. It's almost like reading a book. Here in the United States, we can only read left to right. When you read the other way, it's all gobbledygook. You don't write your sentences starting at the end. You start at the beginning, the 5', and you go to the end, the 3'. Here's another image from the PowerPoint, so you can see a couple connected. Here it goes TAA, but you've got your base attached to carbon number 1. You can see the only ones that are written here are 5 and 3. So from 5 to 3, we attach the next carbon, which is from phosphate to carbon number 5, down to 3, phosphate of the next one, carbon number 5, down to 3, etc. There's a couple of these images here for you to see. Here we've got a dehydration synthesis reaction, basically. Every time we're connecting the 3rd carbon to the phosphate of that 5th carbon, we're going to pull off a water in order to form that bond. So that's a little bit of review from first semester. Okay. Taking a breath. All right, so I think I've covered the nitrogen nucleotide base, the width of the DNA double helix, and the connections between them. So remember, G to C, 3 hydrogen bonds across, A to T, 2 hydrogen bonds across. The yellow that I have here as hydrogen bonding, these are weak bonds, right? It's more of an attractive force from one side of the helix to the other, because this needs to separate so we can access the DNA code inside. But what you do not want to break and separate is from phosphate to sugar, phosphate to sugar. These are covalent bonds that need to hold the backbone securely together. So think about the purpose of those chemical bonds and how they feed into the stability of the structure. Okay, someone's got some little tricks here to remember. PUAG2, so purines is A and G, 2 rings in the structure, or pure as gold, again, purines, the A and the G. So that's a way to kind of remember which ones are which, but that rarely comes up on the AP test, so it doesn't need to be a big stress for you. All right, when replicating DNA, let me look ahead, stand by. Aha, yes. So when a DNA original molecule is going to get replicated, it's here in dark blue. Oops, I've gone up, down. It's going to get separated. We're going to add new nucleotides. So these are getting recycled from breaking down other cells. These are being manufactured by the cell, getting assembled, and we're going to bring in new ones here in the light blue. So in the end, we have what is called a semi-conservative molecule. We have 2 new molecules of DNA where we originally had 1. Each one contains half the original molecule and half a new molecule. This is part of DNA structure where you can use the complementary bonding across to get your other missing side, and then likewise on the opposite side as well. So here are a couple images that you might see on the AP test of ways that it does not work. So here we have like one original molecule, take the original and just make another copy. How do you do that? That doesn't happen. Here we've got the original molecule, and in pieces we're chopping it up and rearranging it with old and new. That would be really hard to keep that in order, so it is not done that way. This way here in the middle it is. You take the original, you separate it so that you have 2 sides that are complementary to each other, but when separated you can fill in new complementary pieces and you continue the process again and again. So the semi-conservative model is how DNA is replicated. Okay, going through here, let's get started. First of all, I want to show you something, but I don't know if I can do it with one hand, and I don't have a dock. The DNA helix being a double helix, I'm looking at my yarn and I'm wanting to like do something with it, but I don't think it's gonna work. Earbuds. Stand by. Okay, I'm back. Didn't leave you. All right, here's my earbuds cord. Imagine this is your DNA double helix. We need to get to the code on the interior. The backbone is simply repeats, sugar, phosphate, nothing unique there. Each nitrogenous base attached to the nucleotide is that unique message that we need to get to, and it's on the inside of this. So DNA, tricky. We have to separate and pull it apart and pull it apart. But just like yarn or these earbuds, as it pulls apart, it's getting a lot of tension down the way. And we need all these enzymes that are going to help us to not only do the separation, but to relieve the tension along the way and lay down new bases. So everybody's got a job here, okay? Let's start with helicase, okay? No, these shapes of these enzymes don't mean anything. I've literally made them look like the letter of the name just to kind of help you out. So definitely watch some of the videos on the online resources. Enzymes are proteins, they're just blobs, all right? But there are specific blobs to their shape and function. So helicase is going to unwind the double helix. So I've got a helicase here moving this way, and I've got a helicase here moving that way. I actually want to add, oh, it is on here. We're going to add the 5' to 3' direction on this upper model because it's not as detailed as down here where we can count. So on the upper strand here, put a 5' heading towards, all the way around, 3'. And DNA, remember, it runs in opposite directions, like this. So opposite that 3', we're going to put a 5' running towards its 3' this way. And remember, each of these little tick marks is like a nucleotide. So this is the 3' of the last one. Way up here, here's the 5' of the first one. Okay? I've colored in dark blue the template strand, just like the original one, and my light blue is going to be the new bases, just like on the PowerPoint, just to keep some consistency for you. So what I've labeled here in this dark blue on my outside 2 strands that go all the way around, that is our template DNA, or the original strand of DNA. Okay, so helicase is going to be unzipping and breaking apart the hydrogen bond. So it's going to break apart here. I've also done little yellow hydrogen bonding across here just to kind of remind you of what we've got down there. So helicase is unzipping. We're going to get a lot of tension along the way, right? If you're untwisting a spiral, we're going to use these single strand binding proteins, and they're going to stabilize the strand as it unwinds. So they are just a couple of proteins that are up and down here on the replication fork that are helping to stabilize. Upstream, as you are unwinding here, you're going to build up a lot more tension this way. So this kind of Teletubby shaped thing is topoisomerase, right? We've got helicase, topoisomerase, primase, single strand binding proteins don't have a ASE suffix, but they are enzymes. So topoisomerase is going to help relieve, that looks like I have white out there, relieves the strain and tension upstream of the replication fork. If you will, I don't have a fork with me, but here is a fork and here is a fork, right? So we are separating out the DNA. So topoisomerase is going to be at either end, helicase is at either end, single strand binding proteins at either end of the 2 replication forks. Still with me? Let's keep going. As we separate, we are going to begin laying down some new DNA strands, but this enzyme, DNA polymerase 3, is kind of a dum-dum. It doesn't know where to start. So primase, what I have in green here, is going to get us started. Primase is the enzyme that lays down about 5 to 10 RNA bases, not DNA, in the 5' to 3' direction to form, you guessed it, a primer. Okay, so that's what I have here. Notice my template strand, like the PowerPoint, is a dark blue. My DNA bases that are going to come in are light blue. RNA, I have chosen a different color, and I specifically have different hashtags here. These are temporary. Primase is laying down just a couple of RNA bases here very closely in 5' to 3' direction, so that DNA polymerase 3 goes, oh, that's where I start. There's a 3' that I can link and move forward in the 5' to 3' direction, because that is all that DNA polymerase can do. With already existing DNA that's there, it can continue and lay the foundation going in the 5' to 3' direction. Okay, so take a look at where primase is. We've got one here, and then we've got DNA polymerase gets a free pass here, basically. If this is 5' to 3', opposite 5' to 3'. As this helicase moves, this DNA polymerase is just going to follow it and keep going on this upper strand. We call this the leading strand. This one has plenty of room and an open road as helicase moves along to keep building in the 5' to 3' direction. Same thing happens here. Primase lays down a little bit, and then DNA polymerase can move. But this is a static model. You can't see this moving. I really recommend watching one of the online resources, because as the replication fork opens this way, we start getting more nucleotides exposed. But we can't like put a DNA polymerase in there. They're going to run into stuff. So we have these little lagging strands that get built, and primase puts down a couple primers, but it's done in these little sections. So primase put one down, DNA polymerase can come in and do a little bit, and another one has to happen because we're opening up this way. So another primer, a little bit of DNA polymerase 3 here. These little fragments that aren't one long continuous leading strand, these lagging strands, are called Okazaki fragments. This is one of my favorite vocab terms from this unit. It's because they're done in little tiny sections, discovered by Mr. and Mrs. Okazaki, I believe. All of this is still continuous, occurring in 5' to 3', and as the bubble gets bigger and bigger and bigger, you're going to end up getting two separate strands, the top one and the bottom one. But over here, DNA polymerase, the only thing it can do is lay down DNA bases, 5' to 3'. Here, where it's laid 5' to 3' and it's trying to connect a 3 to a 5, it doesn't know how to do that. So that's where the L comes in, ligase. Ligase is the enzyme to ligate or glue it together, as you guessed. It's joining the 3' end to the 5' end of any unconnected DNA fragments. So ligase has a lot of work to do here in these lagging strands on these two kind of opposite corners, but it only has one job here from this primer to the DNA polymerase and wherever this one ends, basically. The leading strands are often quite whole. Leading strands built in a continuous piece. Lagging strand built 5' to 3' in chunks as replication fork opens. Okay, I'm trying to see if I have anything else on here. Looking through my phone at this. It's kind of difficult. Otherwise, you get this teacher, and then who knows what you're looking at. So, all right. I'm thinking we're getting most of this. There are a few others that are not pictured on the diagram. So I'm going to come down here. For proofreading, DNA repair, this process obviously does not always work perfectly. Like we were talking about, DNA proofreading enzymes are looking for these kinds of mistakes. Sometimes you could put an A with a C and force it together, and then you have the right distance, and the backbone will not bulge out, but one of them will get replaced, and sometimes it's the wrong one. So we will talk about DNA mutations after midwinter break or whatever the snowpocalypse ends, but that is for the next unit with DNA to RNA to proteins. This is just going over how do we go from one of these to two of these and have everything still be the same. So in mitosis, you're just making clones of the same cells. We need to make an exact copy of what we already have to pass on to the new cell. So to check for mistakes, nuclease is going to cut out any damaged or mismatched DNA nucleotides. So it's going to pull out anything that looks incorrect. And then once again, the simple dum-dum here, DNA polymerase 3 will lay down the new correct base pairs. Ligase will seal the backbone. So nuclease is, in a sense, the nucleotide repairer, right? If some of them are incorrect, nuclease cuts them out. DNA polymerase 1 is going to go back and lay down all the mistakes, not mistakes, I'd say, any of the RNA primers that were laid down by primase. We don't want those ribose sugars, we want the deoxyribose sugar in there. So any of the green little hash marked things I have here, the RNA primers, it's DNA polymerase 1 that is going to swap those out nucleotide for nucleotide. DNA polymerase 2, gotta be honest, don't remember what it is. I have stars here: topoisomerase, helicase, DNA polymerase, and ligase. Those 4 are the ones that are listed on the AP Bio standards of things you need to know for this unit. So you don't have to go that deep into the single binding, the primase, and these extras. I just thought they'd be helpful. But the 4 there you can expect to see on an exam or on the AP Bio test in the upcoming May. That test isn't moving, so let's keep it going, right? Reminder, the DNA replication is happening during S phase. So we've gone through G1, past the G1 checkpoint. S phase is when all of this is happening, and then G2 is checking all the last things before we split. I think that's kind of it of everything that I've kind of covered here. Again, this is scanned in on Schoology. If you weren't able to follow along as quickly, have it filled out, go through it again. Definitely watch some of the videos. Let me see what else I got here on the laptop. A couple of things that you do want to cover that we have not gone over is some of these experimental designs. Messelson-Stahl, 1950, one up at the very top here. This one, the dead bacteria are what is carrying the genetic information. It's not the protein, but the DNA and how it can pass it on even if the bacteria are dead. What else? This experiment, do not be surprised to see it on the exam, about what if I radioactively label the protein, radioactively label the DNA, what is it that's getting into cells? This is a little bit about viruses. This is something, if we have school on Tuesday, Wednesday, we can definitely cover this together. But read chapter 16 to make sure you've kind of exposed yourself to some of this content. Oopsie. I think that's kind of the most of it. Yeah, there's just some other like cool pictures in the PowerPoint. I really honestly think the videos of all of this will be the best resource. Seeing DNA replication happen in real time, moving, all these static pictures are a little bit helpful, but not great when you're trying to explain a very dynamic process. So keep that in mind. Go through the PowerPoint, go through the online resources. I'll post this to YouTube and to Instagram. So we'll see you guys maybe Tuesday, maybe Wednesday. I don't know. My roads look all right over here. So have a great day. Bye.